Hello, this is SabahMusfiah from Department of Electronics Convergence Engineering. Today, I'll be presenting the summary of a research paper titled Ultra-Fast and Recyclable DNA Biosensor for Point-of-Care Detection of SARS-CoV-2 COVID-19. So, this paper introduces a level-free DNA capacitive biosensor designed for rapid and low-cost detection of SARS-CoV-2. The sensing platform is built using interdigitated platinum and titanium electrodes on a glass substrate. These electrodes detect DNA hybridization between the SARS-CoV-2 target sequence and a complementary DNA probe. Unlike traditional methods, this approach does not require labels, enzymes, or complex preparation. To verify the sensing performance, the authors used FTIR, contact angle measurements, and capacitance frequency analysis. The device showed a strong capacitance change of about 2 nanofarads when detecting the conserved RTRP gene region. It also demonstrated high sensitivity around 0.843 nanofarads per nanomolar. Additionally, fluorescence imaging using a dye-labeled SARS-CoV-2 genre further confirmed the selectivity of the biosensor. One of the advantages of this device is that it is reusable. So after EV ozone cleaning, the sensor surface can be used again without significant performance loss. Overall, this developed biosensor provides real-time, cost-effective and high-throughput nucleic acid screening. It can be used not only for SARS-CoV-2 detection but also adapted for other DNA-based disease diagnostics. In this slide, I will briefly introduce the background and motivation for this study. Since the emergence in late 2019, SARS-CoV-2 has spread rapidly across the world. One of the challenges is that COVID-19 symptoms can be non-specific, and the virus can still transmit during the asymptomatic incubation period. For this reason, rapid and reliable detection is critical for controlling outbreaks. The standard diagnostic method PCR is very accurate but has several limitations. It requires long processing times, sometimes up to 72 hours, skilled personnel, expensive lab equipment, and a stable supply to test kits. These limitations highlight the need for faster, simpler, and more affordable diagnostic tools. Point of care biosensors have become a promising alternative because they are simple, low cost, and suitable for mass production. Several electrochemical biosensors have been used for detecting viruses such as Ebola and MERS-CoV, but many still rely on expensive materials or require labeling and amplification processes, which increase complexity and cost. Recently, field effect transistor based biosensors for COVID-19 detection have shown potential, but they still lack the sensitivity of PCR tests. Overall, many current biosensors struggle to detect the very small molecular changes that occur during DNA hybridization. To address these challenges, the authors developed a new level-free biosensor based on interdigitated electrodes. This sensor can detect SARS-CoV-2 DNA through the direct hybridization between probe DNA and target DNA. They validated the hybridization using multiple electrical and physicochemical techniques. The overall goal is to provide a real-time, low-cost, and reliable platform for COVID-19 detection that can also be scaled up for practical use. Here, first, for the reagents, the authors used synthetic SARS-CoV-2 RT-RP gene sequence along with SARS-CoV gene sequences for comparison. They designed probe DNA that perfectly matches the SARS-CoV-2 sequence, while the SARS-CoV sequence intentionally had four mismatches to test selectivity. They also used common laboratory chemicals such as APTase, PBS, sulfuric acid, hydrogen peroxide, ethanol, and DI water. Next is the fabrication and interdigitated electrodes or IDES. A 6-inch glass water wafer was first cleaned and dried, then thin layers of titanium followed by platinum, each 150 nanometers thick, were deposited using E-beam evaporation. After deposition, the wafer was diced into small chips. Each chip had a sensing area of about 13.12 mm square and included 64 IDE fingers. For surface preparation, they used piranha cleaning and UV ozone treatment to create hydroxyl groups on the surface. Then APTase was applied to introduce amine groups which helped attach probe DNA. The probe DNA was immobilized on the surface at 60 degrees Celsius for 3 hours. For target detection, both complementary and non-complementary DNA samples at a concentration of 5 micromolar were introduced to the sensor. The hybridization process was performed for 1 hour at 60 degrees Celsius. After hybridization, capacitance was measured from 10 to 1000 Hz using an impedance analyzer with a 500 millivolt AC signal. Finally, for surface characterization, the authors used contact angle measurements, ATR, FTIR, and fluorescence microscopy. These methods confirmed successful surface modification and validated DNA binding on the sensor surface. Here first regarding the operational mechanism, the probe DNA was designed from the SARS-CoV to RT-RP gene, while the SARS-CoV sequence with 4 mismatches was used as a negative control test specificity. The IDE sensor contains 64 interdigitated fingers, giving a relatively large sensing area of about 13.12 square micrometers. The sensor surface was prepared through hydroxylation followed by APTS coating and finally probe DNA attachment. Only when complementary target DNA binds to the probe DNA does double standard DNA form, which leads to a measurable increase in capacitance. Next is the physicochemical analysis. Contact angle measurements confirm that the surface was successfully modified. The angle was less than 3 degrees after hydroxylation and around 39 degrees after APTase coating, showing that each step worked correctly. Here, capacitance frequency tests showed that the strongest and most reliable signals appeared at 10 Hz, with capacitance consistently increasing after each modification step. Finally, for sensitivity, specificity, and recyclability, the sensor showed a strong and linear response to SARS-CoV-2 complementary DNA with a sensitivity of 0.843 nanofarads per nanomolar and a detection limit of 10 nanomolar. There was no significant response to the mismatched SARS-CoV sequence, which demonstrates excellent specificity. The complementary DNA produced a clear capacitance increase of around 2 nanofarads. Importantly, the sensor remained stable even after 5 reuse cycles. After each cycle, piranha cleaning and UV ozone treatment effectively regenerated the surface. Overall, these results show that the proposed biosensor is sensitive, selective, and reusable, making it a strong candidate for rapid COVID-19 detection. In summary, the authors developed a fast and low-cost capacitive biosensor for detecting SARS-CoV-2 DNA. It showed a clear capacitance increase only for the correct SARS-CoV-2 target, providing high sensitivity and specificity. Hybridization was confirmed through electrical and optical tests. The sensor can also be reused multiple times after simple cleaning, making it practical for low-resource settings. Overall, this biosensor has strong potential for point-of-care COVID-19 diagnostics and future commercialization. Thank you. That's it for today.